Very good morning, friends. Myself, Dr. Naval Singh Pudawat, Assistant Professor in the subject of Botany from Morishwar Arts, Science and Commerce College, Bhokarwal. Friends, we are studying the genetics and in that we are touching the aspect gene interaction. We have gone thoroughly through this chapter, that is the gene interaction, allelic gene interaction as well as the non-allelic gene interaction. While studying this non-allelic gene interaction, we have to see that there are four types of the non-allelic gene interaction. Now, which are those? That is the complementary gene action, epistatis, dominant, recessive, while duplicating factor. Then the last one, that is the supplementary gene action. As in the complementary gene action, we come to know that the ratio we find, that is 9:7 in the Mirabilis jalapa plant. Now epistats, that is dominant epistatis, recessive epistatis. Well, in the summer squash we find out the color, that is the ratio here of the color of the summer squash, that is the 13:3:1 in the dominant epistatis. Recessive epistatis. In recessive epistat, we study the coat color of the mice, and in that we come to know that is the 9:3:4 ratio of the agouti black and the albino. We're talking about the duplicating factor. What is the duplicating factor? A single character that is controlled by two independent genes which express a seed character. With regards to the shape of the seed character, the other gene is present or absent. This is the indicator to know the gene is present or absent. It was observed in the shepherd purse, and the seeds were selected of a two type of a two character, that is the triangular seed and the top seed. When we go for the genotype of this triangular and top seed, we come to know that the capital double T and capital double D, that is the genotype of the triangular seed, whereas of the top seed, the genotype is the smaller t double and the smaller d double of the top seed. When these are crossed, and what we get in the first filial generation, that is the triangular seed. But now the genes or the genotypes are somewhat complex, different than the parental triangular seed. While we move forward, they are allowed to self-pollinate, and what we get in F2 generation, we find that is the triangular shaped seeds were 15, and top shapes were 1. That is, well, we got the ratio, that is duplicating a factor of a. Next, that is the supplementary gene action. What is the supplementary gene action? What is this phenomenon? Two independent dominant gene which interact in such a way that one dominant gene produces its effect. Whether the other dominant gene is present or not, but when the second dominant gene is added to the first, a new modified characters are produced. Well, gloom color of millets is the best example of the supplementary gene. In supplementary gene action, two independent dominant gene which interacts in such a way that one dominant gene produces its effect, whether the other dominant gene is present or not. But when the second dominant gene is added to the first, a new modified character produced. So the best example of the gloom color of millets, we have phenotype purple into brown, and the genotype is as shown in the slide. Gametes are PQ, PQ, vice versa, the small one, bigger letter. In F1 generation, we got the complete different color, that is the red purple. Now we allow to undergo the F1 generation for the self pollination. What will we get in the F2 generation while this red purple is crossed with the red purple? Now in the F2 generation, while in Punnett square, what do we see in the Punnett square? From the left hand side, what we see that the female gametes, and from the right hand side, that is the male gamete. While capital PQ crossed with the capital PQ, we get the red purple. Capital PQ with the capital P is crossed with the, then we get the red purple too, but the genotype is somewhat different. In the same way, the phenotypic ratio that we get is to 9 is to 3 is to 4 in this experiment. Now what is the 9 is to 3 is to 4? That is red purple, purple and brown one. Friends, while looking out this gene interaction, while looking out the gene interaction, what we come to know, that is, according to the Mendelian or Mendel, each and every character was controlled by a single gene. Later, it is known as gene interaction. This can be studied under two different types. What are they? Allelic gene interaction and non-allelic gene interaction. In regards with the allelic interaction, it is explained under two different examples. Whereas in non-allelic gene interaction, which is explained under four different types, such as complementary gene action, epistasis, duplicating factors, supplementary gene action, and it was given by a suitable example. So this was what about the gene interaction summary, and well, one has to repeat this for understanding in our next lecture or in our next topic. We'll be dealing with the sex determination or the sex chromosome. Thank you.